Bio, where we develop biotools to simplify complex biotherapy development. So gene and cell therapy has been at the forefront of R&D since the 1980s, and the first therapy was only approved in 2017. But there's been a lot of drugs in the clinic right now, currently, and they all have generally a monogenic focus, which is a single target, single gene therapy that can really address a minor set of diseases, but simply cannot address the complex multi-gene diseases that kill most people in the United States and the world. And the question is, why are there no complex therapies? Where are they? And it turns out that they're not even in the lab, because there aren't any tools available for researchers to even begin thinking about designing, let alone creating, multi-target therapeutics. And so, to put this into context, here are all the papers that have been published using legacy genome engineering tools. It's a disappointingly small number, but these technologies are not bad. They're just really difficult to use. It requires a lot of expertise. Then a new kid on the block comes along. We all know CRISPR, and just goes through the roof on impact. Because CRISPR is exactly what happens when a needed tool is made easy to use. And so at Osprey, we've taken that to heart and have developed a platform and products that are even easier to use to do something that's far more complex, making multi-target, multi-gene therapeutic candidates. So we're introducing the clone card. Oop, that doesn't look that good, but those are supposed to be QR codes. Introducing the clone card, which is the first accessible multi-gene construction tool on the market for research and clinical development in cell and gene therapy, powered by our Bird of Prey platform, which is a very simple platform. Every gene looks like the base vector on the left, with different modules that researchers can cut and paste, swap out to build multi-effectors with multiple proteins in one gene, or multi-genes with full transcription units, each with multi-effectors within it. So like lots of possibilities. I mean, it's nearly infinite. And so the clone card is how you wield this power. These 8 gene cards have combinations of genes that are relevant to certain disease classes or functionalities. Researchers will do the cut and paste with Bird of Prey, build a multi-gene, much faster workflow, and the cost is so much less because you're testing multiple things at once. You're not doing one protein, one pathway like they do today. It's very much a quick process for multiple things at once, and it does not require expertise. That's the big thing. So our current and future offerings allow us to address several segments in biotools, which is a very large market, very dynamic, from basic research to preclinical activities. We're focusing specifically first on the 5 billion gene and cell therapy subsegment, or discovery subsegment. 5% capture, this would be about $250 million in revenue, which we believe is attainable. Our competition is essentially anybody who provides DNA to customers, but we are by far the only platform that provides the multi-gene building system. And we've also priced everything to be essentially the lowest on the market to make access that much easier. And if we look at our revenue projections, the reason that is because we have 3 different sets of cash flow. We have clone cards, we have recurring tools like reagents and kits that you need to use if you want to use the clone card, and then licensing opportunities downstream. Although bootstrapping is feasible, we are raising a $1 million angel round to kind of supercharge everything that we're doing, so increase our product line and then execute a really aggressive sales and marketing plan that is already in place. Our team has a lot of experience. This is not our first rodeo. Biotools has been a specialty for our board, for example, and together we would like to ask you to help join us and bring the world to a new future where we can actually think multigenic. Thank you.